Hi, I'm Dr. Elise Wright, and I work as a postdoctoral research associate in Dr. Zoe Waller's lab in the School of Pharmacy at UEA. The aim of this video is to explain our recent research paper to a general audience. It has just been published in Biochemistry. The paper is open access, so absolutely anyone can read it. The links to the paper are included on the YouTube page. The title of the paper is Substitution of Cytosine with Guanylurea Decreases the Stability of i-Motif DNA. This work is the result of a collaboration between both the Schools of Pharmacy at UEA and the Beckman Research Institute and Division of Urology at the City of Hope. So where does the story start? This story starts with DNA, or rather the sequence of DNA, the instructions for life that are spelt out using 4 building blocks we call bases: adenine, thymine, guanine, and cytosine. The DNA sequence is like a kind of blueprint, with all the information you need to build a person, just in the same way as you would have a blueprint for making anything else, even a Millennium Falcon. But there's all kinds of additional information on DNA, like special chemical markers. These are extra pieces of information all over our blueprint. The position of these markers is important, and it needs to be the same every time our DNA or blueprint gets copied. This is important because there are drugs which work by disrupting how the pattern of chemical markers is maintained. Decitabine is an example of this kind of drug. It is used to treat acute myeloid leukemia, a cancer of the blood and bone marrow. It works by stopping markers from being added to DNA. It rapidly breaks down in water, and as humans are mostly water, we're very interested in its breakdown product, 2-deoxyriboguanylurea, which we call Guanylurea. Now let's jump back a bit. Do you remember the 4 building blocks which make up DNA sequences? In particular, cytosine? This is what the chemical structure of cytosine looks like. This is the base that the drug decitabine mimics, as you can see, which, as we know, then gets broken down into Guanylurea. We are interested in the base C and any of its mimics because sequences of DNA which contain lots of this base can form unusual DNA structures. We call these i-Motifs. These look very different to the typical double helical structure first proposed by Watson and Crick in 1953. So what happens when Guanylurea gets into these i-Motifs? Could this be part of how our chemical marker pattern gets disrupted? We try to answer these questions by looking at the DNA and how stable it is. We use a technique that can measure the stability of DNA. For example, if we take an i-Motif and slowly apply heat, we can find out how much heat is needed to make it unfold. When we do the same thing with the Guanylurea in the i-Motif, we find we need less heat to unfold the structure. We can do the same thing with changing the acidity of the DNA. This allows us to understand the stability in a different way. And again, once we introduce a Guanylurea, the i-Motif is less stable and unfolds sooner than the cytosine-based i-Motif. So what does this mean? Well, this was quite the surprise. Guanylurea looks so much like cytosine, it was expected to behave in a similar or better way when it was all folded up into an i-Motif. This research helps us understand more about the breakdown product of decitabine, Guanylurea. It also tells us that the interaction between this drug, i-Motifs, and the chemical markers on a person's DNA is a whole lot more complicated than we anticipated. This work has been supported by the Biotechnology and Biological Sciences Research Council, or BBSRC. If you have any questions, please feel free to ask either by tweeting Zoe Waller or by email. Thanks for watching, and tune in next time for more science awesomeness.